The next talk is by Ryan Kudenkunz from the University of Arizona.
Okay, thank you everyone for staying as always. So GHIST is the Genomic History Inference Strategies Tournament, and this is really a community-driven effort that really depends on your engagement and your participation. So if you've seen that scary message that flashes up about posting things on social media, I am not only giving you permission to post on social media about this, I am begging you to do so. So please, help spread the word, because we really need engagement to make this successful. And to motivate this tournament, which is focused on population genetic inference, I want to start with a metaphor that was suggested by Richard Durbin, which is to think about analyzing population genomic data like how we think about the parable of the elephants and the blind men. Just like the elephant, population genomic data are complex. They come from a biological process, so they're messy, and they aren't going to be captured by any very simple parametric model. But we, as the blind men or the population geneticists, tend to look at particular aspects of the data. So one of the men might grab the trunk and say, oh, the elephant is like a snake. And that might be someone in my group who is using the joint site frequency spectrum to learn from the genetic data. Whereas another one of the blind men might touch the tusk of the elephant and say, oh, the elephant is like a spear. And that might be someone looking at pairwise coalescent times. And we have lots of different ways of looking at the elephant. Segments of IBD or modern machine learning methods based on summary statistics or haplotype matrices, but nothing is really capturing the entire elephant. None of our methods really encompass all the complexity that we see in population genomic data. So we'd like to get better, of course, but there's a very practical question for people who want to use the methods we develop, which is, how do they know what to use when? Now, I am in a room of methods developers. I'm a methods developer myself. When we publish our papers, we publish benchmarks and tests, right? We evaluate our methods. But I want to put us all on blast a little bit and say that I don't trust the benchmarks I put out. I don't trust the benchmarks you put out, because even if we have the best of intentions, when we're evaluating our own tools on data we created, there are lots of opportunities for even unconscious biases to come in. We might be cherry-picking the scenarios we're testing because we really want to focus on where we know our method does well compared to competitors. We developed our method. We know exactly how to fine-tune it and how to get it to perform best, whereas if we're comparing with some other methodology, we might be just using the default parameters. And lastly, if we're testing on data that we generated, we know what the ground truth is that we simulated, and essentially we can peek underneath our blindfold and see what the elephant looks like. And so any sort of inferences we do, we're often going to be biased. It's just human nature that you want to do better with your tool, so you're going to be a little biased knowing what the result is. So this is a real hard challenge, and how do you do good benchmarking? Ideally, you would have it be done independently, but that's very hard and takes a lot of effort on groups that aren't necessarily going to be the most rewarded in science. So one way we can avoid particularly this peaking problem is if we hide the truth. Right? If the people running the benchmarks don't know what the elephant looks like, then there's much less opportunity for massaging the results to make them look better. And one way to do this and really engage the community and get a diversity is a competitive format. And many of you might be familiar with the most famous competition in computational biology, which is the critical assessment of protein structure prediction, which has been running for over 2 decades and has really delivered amazing results. So shown here is a measure of how good the predictions are, with better being up top. And on the x-axis is a qualitative measure of how difficult that protein structure people expected to be predict. So going from sequence to 3D protein structure. And each line here corresponds to a different year of the competition, and you can sort of see this slow progress but then there's a few big jumps in performance. And as you may well be aware of, this black line here, that's AlphaFold 2, right, which has revolutionized biology and made many, many, many millions of structures available to us, and really changed how not only computational biologists do their work, but also real in-the-lab molecular biologists who are thinking about this every day. And I think if you talk with the DeepMind developers, the existence of this competition played a real important role in driving the field forward and motivating them to become part of it. So I want to humbly try and make GHIST like CASP eventually, right? We want to build a series of competitions that will engage our community to help us develop our methods, push science forward, and eventually try and engage others outside of our community to bring in their ideas too. Now, I keep saying the word community. I've been the person who's been doing the organization and the technical bits behind the scenes, but I really depended on a lot of advice from the design committee, trying to recruit people who could help give me good ideas, a couple of whom are in the room, Peter and Adam. Katie Lauderhouse, I thought, had some really key insights. In particular, it's very tempting to be very ambitious and make things very complicated and hard. And she had the really great advice that, especially in the first few years, you've got to make things straightforward and easy for people to participate. And Andreas was really instrumental in helping us get the word out for particularly the first year of the competition through his connections with SMBE. The behind-the-scenes, nuts and bolts implementation of the competition was really managed by Travis Struck, who is a staff scientist in my group. And we really benefited from leveraging the Synapse platform that the Sage Bionetworks group has put together that provides us leaderboards, provides us easy ways to distribute test data, to evaluate test competition entries, and just makes the whole process run so much more smoothly than if we had to roll our own. So we've had 1 year of competition so far. What did it look like? So we started off in July at SMBE, where I ran a kickoff meeting and ran a workshop where people could download data, do some real quick inferences, make their first submissions. And we ran the competition until November. So we had about a 5 or 6 month window. And this is really, the goal here was to capture people in the summer when we're all meeting and interacting at meetings, try and build some hype, and then to extend the competition window into the fall so that new graduate students, new trainees could come in and hopefully apply some of these methods as part of their training, right? This is a great opportunity not only for the experts to evaluate sort of the bleeding edge, but if you have a new student joining your group, here are some data sets that are easy to access that they'll be able to take whatever tools you love to use in your group and just try them out, right? We're trying to make this quick and easy and really accessible. So we ended up with roughly 60 participants. And again, I'm going to keep saying these words, in the interest of keeping things straightforward, for the first year we just had 4 challenges of inferring demographic history. Not because I think it's the most important problem in population genetics, but just because it's something that my group knows well. There's a lot of tools out there. There's a lot of opportunity for people to dive in. So this year, the data we provided were error-free data provided as VCF files. So we, again, making things simple, making the easiest possible inference we could do. In the future, I could envision distributing reads or error-prone data to really test the boundaries of our methods. And we asked competitors to submit estimates for a few numerical parameters per challenge. Just again, to keep things pretty simple. In the future, there is the capability to ask people to submit things like distributions of coalescence times or almost anything you could imagine. We could have it, if you're really ambitious, you could even ask people to submit pipelines as Docker containers and run them on unseen data. But given that very simple input that we asked the competitors to give us, we had a very simple scoring metric, which was just a relative root mean squared error between the submitted values that the competitor submitted and the actual true simulated values. And this showed up on a leaderboard. And I think that's an important aspect to keep people motivated to see, okay, how well did I do in my last submission? We also asked competitors to submit a brief write-up of their procedures so we could understand what people were doing, and there were some really interesting stories there. And the carrot for this year was that all the top competitors are being invited as co-authors on the manuscript describing the work. And I should mention that, of course, all the data we're providing here is simulated data. Our field, at the present, we lack real biological ground truth data that we know the entire evolutionary history. But we do have these really powerful simulators that can introduce complexity and biological realism that no inference method can currently capture anyways. So I'm comfortable using simulated data at this stage in our evaluation of the field. But turning back to those top competitors, here are some of them, some of whom are in the room: Aaron Ragsdale, Andrew Vaughn, who is a graduate student with Rasmus Nielsen at UC Berkeley. Austin and Dylan are from Dan Schreider's group at UNC Chapel Hill. Eilish and Toby come to us from the University of Sydney. And actually, their input was some of the most useful, because I would say they are the least experienced and the most newbie to this sort of inference. And so they had really great insight into what was confusing, what made it hard to participate, and what makes it hard to use the tools that we all produce. And lastly, Ekaterina Noskova ran a really big international team, all using the tool that she's developed, GADMA, which is an interesting sort of meta tool for demographic history inference that provides access to a bunch of inference engines like Daddy and Mommy, and also on top of that can systematically build more and more complex models. So we started simple, as always. The first challenge was simply to infer. We gave them data with a bottleneck model and asked people to infer how long ago did this bottleneck occur and how severe was the bottleneck. And so shown here are many of the submitted entries. And on the left here, you can see the distribution of scores, which is quite wide. On this relative root mean squared metric, orders of magnitude difference. And if you look at the XY plot here of time of the inferred decline versus the fraction, the severity of that decline, where the lines show the true value, you see that everyone inferred that, yes, there was a pretty severe decline. There was some scatter in the inferences of the time, but some people did really, really well. That there were a lot of entries that were very, very close to the actual true simulated values. So let's zoom in there. And there's a really interesting story here. So here we see some of the best inferences. And I want to highlight a couple. So in particular, one competitor developed his own approach using TS-kit to simulate the site frequency spectrum and using KL divergence to measure the difference between the data and the simulated spectra. And their first guess was pretty good. But then they were able to refine that guess in their second submission to the competition. And I think then this particular individual was, you can either call them clever or nefarious, because they thought we were humans generating the data. We probably use round numbers. And we're able to then optimize not the population-genic data, but their position on the leaderboard. So they were able to get closer there and then do a very simple binary search with their fourth entry to try to the right. That got a little worse, and they could go to their fifth and final entry to the left, and wow, they did so good. And I am going to call out Andrew Vaughn, sitting here in the third row, as the competitor who broke the system. Wave to the crowd. Oh. So our second challenge was to try to up the complexity just a little bit. We had a split isolation model, again, a pretty simple population genetic scenario, no gene flow between these populations, and we asked people to estimate the contemporary population sizes in the generations since the split. Again, given the simplicity of the scenario, you might imagine that people were able to do pretty well on this. So Andrew Vaughn's SFA-based inferences were pretty good. From the Schreider group, they developed a machine learning approach using simulations to generate vectors of summary statistics, and they also had quite accurate inferences. And our newbies actually did pretty well using the command line interface to DADI. But these are not actually all the top scores. Again, Andrew was able to metagame us and top the leaderboard. So for the third challenge, we wanted to mix things up a little bit and start to break some of the assumptions built into our models. So the scenario we posited here was one of secondary contact. We have a mainland population and an island population that diverged sometime in the past. And then they recently became back into contact and started, and gene flow started between them. And we asked competitors to estimate population sizes, contemporary population sizes, the timing since the split, the timing, the onset of gene flow, and the rate of that gene flow. And you notice here that we added a whole bunch of extra wiggles in the demographic history with the idea of making things a little bit more close to the complexity we might expect in natural data, which of course aren't going to be simple exponential growths or simple constant population sizes. And what you'll see here is that the scores are much worse. No competitor really nailed it, I would say, and there's a pretty wide range of things that they did accurately versus inaccurately. And Andrew was not able to metagame us on this competition. The best result here comes from the GADMA team. And I think this is really highlighting the strength of this tool because GADMA builds the parameter model as you go. And so you start off with a simple model, and then it'll propose almost like an MCMC sort of step. What if we add exponential growth or a size change or add complexity automatically? And I think that helped them capture some of these wiggles we put in without having to pre-propose them as a prior. And as our fourth and final challenge, we wanted to switch things up a little bit, so we put a challenge out to infer parameters for an admixture model. So we had a pretty complicated set of populations and demography, but we really just wanted pretty simple inferences about what the admixture proportions were from these two archaic groups into the modern populations and when they occurred. And the goal here was to switch up the task a little bit and try and get some of the more specialized methods like Priya's groups developed to be applied here. We didn't attract that community, I'm a little sad to say, and so people actually did quite well just relying on the site frequency spectrum, but it would be really interesting to see, I expect that the people who really think about this problem hard and work with track lengths would have knocked it out of the park. So what is this year's competition going to look like? So one of my goals is to widen the variety of challenges. We'll still do some basic demographic history because that remains sort of the cornerstone of a lot of what we do in population genetics. And we really want to get people using those cutting edge methods, right? No one used ARGs in the competition this year. We need to see some ARG inferences next year. If these methods are so great, prove it. We'll also be able to throw in additional complications. Demographic history, we can add in background selection now that the standard PopSim consortium has built those really powerful frameworks for simulating those data easily. I'm talking with Andy Kern about adding a task of detecting positive selection because that is also an extremely popular task in population genomics, and we have lots of methods to evaluate. And we'll be kicking off at the Evolution Conference this June in Athens, Georgia. So if some of you are there, I think my workshop is during lunch on Monday. So come on by. You can register ahead of time, or you can just drop in. It'll be a hoot. So in summary, I think the first year of this competition has really demonstrated feasibility, that we can do it. Technically, there were little bugs along the way with, AWS instances crashing, but we worked through those. I don't think we have definitive results yet, but at least in this first round of competition, the demographic history inferences were led by a variety of methods based on site frequency spectra. I think that those are the most common tools nowadays. I don't necessarily think they're the best nowadays, though. And Andrew really gamed the leaderboard in a really impressive way, which leads us to think about how do we reform the competition structure to discourage that, while also giving people the opportunity to make multiple submissions and iterate. And GIST2 will have more task variety. Like I keep saying, this is only going to live and die based on your participation. So please contribute your entries and your ideas. During the question session, I want you to scan this QR code and sign up for our mailing list. I will notify you when the competition is live. I promise not to spam you. And I'm really open for both your questions now and your comments that are not really a question, but I want to hear those too.
Thanks, this is really cool. So I guess for the current form, you are giving people like a baseline model and then ask people to estimate parameters within this model. But I guess like in a lot of literatures, people argue about like whether this model or that model like evolving like one ghost introgression or two ghost introgressions or pulse strength. There is a huge element of like just trying different models and then testing for their fitness. So I wonder, like, what's your thoughts on designing like new competitions which will merit goodness from different models without telling people a baseline just to optimization on it?
Yeah, so we didn't give people, this is the model we should fit. We gave sort of a verbal description of, the squirrel example was like, oh, the canyon formed that separated them, and we want to know when the canyon formed. And, it was up to the users to figure out what sort of models they wanted to fit. Like, I expected people could also use PSMC or MSMC and stare at those plots and, make a qualitative observation too. So we're trying to make that accessible. It is a challenge as to how much do our verbal descriptions steer people certain ways.
Yeah, but you have to measure your goodness by defining that like error term. But if you have unequal number of these parameters, how would you assess the goodness?
Yeah, that's a good question. I think it's really the way we thought about it was, what are the headline numbers that people might want to put in a paper that like Priya has an age for a time for the admixture event, right? That's something that is going to be picked up by the press. They're not going to report on a distribution of coalescent times or something like that. If we really want to access the experts, we can do that. It's just more technically difficult.
Hi, very nice talks. I'm not at all from the field, but my question is like, have you thought about it making like very easily accessible from people from like machine learning, where in NeurIPS they have like competition tracks where you can like engage a very broad community to kind of pay attention to this challenge. I think like a couple of years ago there was a single cell data challenge in NeurIPS.
Yeah, I think that is definitely where we'd like to go. It's not clear to me what the best way to do that is. One way to do it would be that we could provide training data in the sense of lots and lots of simulations under a huge variety of scenarios and say, here's your training data, here's some real data, use whatever machine learning method and cleverness you want to get us there. That's one way I've thought about doing it, but again, I'm open to suggestions about what is the best way to really make things most accessible as possible.
Hi, thanks. I think that's super cool. Thanks for doing it. I was thinking about the loss function. The RMSE is obviously quite natural in ML, but it doesn't feel like the right thing here, in the sense that you can be. I think basically what demographic inference is density estimation, particularly in the one population case. So the right metric is actually just measuring the difference in density between the truth and whatever they submitted. And so you can summarize that as a number still. It's not RMSE, but it's still something that's useful. And I think that kind of gets to Yoon's point about model misspecification. So the model will always be misspecified in real data. So having something that is not sort of dependent on the particular coordinates that you chose to parameterize the model, but rather is agnostic to that, I think maybe is a cool direction to consider.
Yeah, I agree completely. I'd love to pick your brain about what you think the best densities are to compare. And I think there's two tracks, right? There's the inferences you do to tell the ecologists or the biologists or the archaeologists what you wanted some number that they can compare with their data. And then there's the inferences you want to do because you want to create a background null model and doing some other analysis or something like that where you really care about, say, a distribution of coalescence times in your null model.
Thanks. Hey, Ryan. Yeah, great talk, fun stuff. So I have two non-question comments. So the first is about what you were just talking about with trying to get more machine learning people on board and maybe supplying them with lots of training data, which I think is a great idea. But one thing to that maybe we should try to impart to any of those folks who express interest in joining this competition is that a lot of the important work there might be figuring out what your training data should be. So you could use our training data, but if you can invest some time into learning how to generate your own, that you could get a lot of gains out of that. But yeah, I think it's a great point, a great idea. We want to bring more people in. Hopefully someone like DeepMind will come in and blow up our entire field and solve all the problems and we won't have to do it anymore. Yeah. And the other thing is just to clarify a point you were making, I think you were suggesting that the more people who participate in this, the faster our methods will improve, the better they will get. And if we all actually want to see things progressing, we should participate. Is that?
That's my feeling, and I will say my experience is going to conferences and talking to people, and they say, "Oh, that's awesome! I'll definitely participate." And then, of course, the rubber hits the road, and we all have lots of other obligations. And I want to engage people to say, try it out, or at least get your students to try it out. They have more time than you do, probably. So twist their arms a bit, and they'll have fun. They'll learn things. They'll earn some authorship, probably. And I think next year I'm going to buy some gift cards or things like that to really, help them buy some books or coffee at least.
Yeah, and I just add, they don't have to spend that much time on it. You don't have to, use up a ton of your effort, ton of your students' effort. Just a little bit of time can make a big difference, I think.
Yeah, I agree completely. Yeah.